I think we have like 30 seconds. Okay. Pam's going to make an announcement and then I'll introduce you.
Good afternoon. Let's be getting our seats. For those in the room, if you have a cell phone, if you could silence it, it would be greatly appreciated so it doesn't go off during the talk. Muters or people on Zoom, you are muted. I always do that backwards. You are muted, and so please stay muted. When questions and answers at the end of the presentation, put your questions in the chat and I'll monitor the chat and we'll ask the speaker the questions. And so Mary Chrisman is here to introduce our speaker for today.
Hopefully this is on. We are very honored today to have Dr. Pam Soltis. She is an amazing background. Wait till you hear this. She's a distinguished professor and a curator at the Florida Museum of Natural History at UF and director of the UF Biodiversity Institute. She is director of research for iDig Bio, the National Center for Digitization, did I say that right? Of biodiversity collections. She got her BA from Central College in Iowa and her PhD from the University of Kansas. Her research focuses on the patterns and processes of plant evolution, spanning from the genomic, so DNA, all the way up to the landscape scales, with applications for biodiversity assessment and conservation. She has developed outreach products that combine science and art, emphasizing the tree of life. There's so much to tell you. She served as the president of the Botanical Society of America, the Society of Systematic Biologists, the American Society of Plant Taxonomists, the International Society for Phylogenetic Nomenclature, the Academy of Science, Engineering, and Medicine of Florida, and is secretary of the board of directors of the American Institute of Biological Sciences. She has emphasized scientific literacy, like this, opportunities for early career scientists, scientific communication to the public, and diversity in the scientific workforce. She's a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. Now listen to these prizes. She's awarded the Dahlgren Prize in Botany, the Asa Gray Award, the Stebbins Medal, the Botanical Society of America's Merit Award, the Darwin Wallace Medal, at least they recognized Wallace, huh? All jointly with her husband and collaborator, Doug Soltis. The Southeastern University Research Association Distinguished Scientist Award, the University of Florida Teacher Scholar of the Year, the Southeastern Conference Professor of the Year in 2023, Teaching and Mentoring Awards from Washington State University and the University of Florida. So, with no further ado, I would like to introduce Dr. Pam Soltis, who will today be talking about evolution in flowering plants. And I have a question for her already. This is the classic, which came first, the insects or the flowers?
Oh, thank you. Oh, I'll use this one. Thank you. Well, thanks so much, Mary, and thanks to all of you for coming. It's really a thrill to be here. I've been here a few times in the past, but it's been a few years, so it's really nice to be back. And I'm really excited to share some of the work that we've been doing over the literally decades as we've been studying the diversity of flowering plants. And so I'd like to talk about speciation and also flowering plants. But in order to do that, we actually need to have some context and maybe a few definitions. So first of all, we'll just start out with this word phylogeny, which I will probably be using pretty frequently in spite of myself. But this really just means the evolutionary history. And we can convey this as a family tree, maybe in a structure such as this, or in some cases, if we have a lot of species, they don't fit very well on a diagram like that. And so we turn them into a circle. So it's basically a family tree like we all might have of our own families, but we can either look at them like this, we can turn them on their sides, we can turn them upside down. They all tell us the same thing. Or if we're scrunched for space, we can make them into a circle, where the origin of the evolutionary tree, or phylogeny is somewhere here in the center. And we have these branches that all convey relationships just like this one does. And what this is showing us is that taxa B and C are closest relatives or sister species, and that their closest relative is taxon A. So that's sort of one of the basic principles of what we'll be talking about today. So I want to share with you some of the work that we've done on constructing and interpreting this large scale phylogeny or evolutionary tree of plant life, but also some of the mechanisms that have generated that diversity. So this is just another example here, again in circular form, but with a bunch of photos around the edges. And this one actually shows all of the so-called green plants, which go back to the algae and then show the relationships as organisms, plants in this case, proliferated through time. And it also shows here the red algae, which are the closest relatives of all of the green plants. Now, when we're talking about the green plants, we're talking about a huge group of organisms, 500,000 species, probably more. This is about what we have named. And they trace their evolutionary history back nearly 1 billion years. So this is one of the major branches of the tree of life, and certainly represents a huge contribution to species diversity on our planet. If we focus just on the angiosperms, I should probably go back a little bit. So let's see. So here, we've got red algae, green algae. We start getting some ferns and their relatives. We get to the conifers and other gymnosperms. And then the flowering plants start right there, and they go all the way around to the end. So most of these species are angiosperms or flowering plants. And I'll use those terms interchangeably. And this is just a really nice depiction of some of these relationships within just the flowering plants, taken from illustrations from Fairhope Graphics. So out of all of these, about 500,000 species of green plants, by far the majority are the angiosperms or flowering plants. About 400,000 named species here, and they go back in the fossil record only about 140 million years. Now, this is in stark contrast to some of those groups like the ferns and the gymnosperms, which go back over 300 million years. So there were a bunch of different groups of plants that got started shortly after plants made it onto land, but it was quite a bit later when the angiosperms actually arrived. And when the angiosperms originated, the fossil record says 133 or so million years ago. Molecular evidence says maybe a bit older. This still is quite recent in terms of geological time. And what this means is that this vast diversity of flowering plants has originated and become established and become dominant on the face of the earth in a very short, geologically speaking, period of time. So the angiosperms then represent one of the greatest terrestrial radiations, and they are ecologically dominant, and you can find them essentially in all terrestrial environments, and even in some aquatic environments as well. So the angiosperms have managed to permeate essentially all habitable parts of the globe. Now, just to put the angiosperms into perspective with other groups of organisms you might be familiar with, let's think about some vertebrates. There are about 66,000 species of vertebrates, not 660,000, 66,000 species of vertebrates. There are about 11,000 species of birds, about 6,000 species of mammals. So the number of angiosperm species absolutely dwarfs these very recognizable and commonly studied and appreciated elements of the animal part of the tree of life. Now, of course, if we're thinking about insects or so, well, the insect numbers dwarf the plant numbers. And if we start thinking about bacteria, well, the bacterial species, we don't even know how many there are. We don't even know what a bacterial species really is. But there are a lot of lineages of bacteria that basically put all of the rest of the tree of life, the plants, the animals, and the fungi to shame. Okay, so that's a little bit of an introduction to our cast of characters, the angiosperms or flowering plants. So if we want to think about how all of these species originated, then we need to start considering what sorts of processes might drive the origin of new species. And so if we go back to this very simple phylogenetic tree, we can then start asking questions about what happened here that drove the split between B and C, and what happened back here deeper in time that drove the split between A and this branch that eventually became B and C. So, of course, Darwin addressed this question, and this is a beautiful quotation from The Origin of Species. And you can probably all see that, but just in case, I'll just read it. As buds give rise by growth to fresh buds, and these, if vigorous, branch out and overtop on all sides many a feebler branch, so by generation, I believe it has been with the great tree of life, which fills with its dead and broken branches the crust of the earth and covers the surface with its ever branching and beautiful ramifications. We don't write like that in science anymore. This is beautiful. It's beautiful writing that depicts the overall process of the origins of new species and the extinctions of others. So let's go back to our tree and start to think about what sorts of processes might lead to the origin of new species and the speciation process. So of course, in order to be able to do that, we also need to make sure we're all on the same page with some terminology. So we're going to address three what seem to be really simple questions. What is a species? I'll tell you, not a simple question. Biologists continue to argue about what a species is. And for good reason, because there are reasons why it actually really matters. The second question, what is speciation? And then the third, what processes can lead to speciation? So let's address this first question. What is a species? Well, as I mentioned, there are many definitions, and people tend to disagree and disagree vehemently with others about the best definition of a species. Now, Walter Judd, a former colleague of ours from the biology department and the Florida Museum, was an expert plant taxonomist. He still is, but he doesn't live in Gainesville anymore. He's moved to Minnesota to be closer to his children and grandchildren. But he wrote a beautiful textbook. And in this textbook, they provide a very simple definition of a species, a basic grouping of organisms. That is one of the few definitions that nobody has an argument with, because basically what we want to do is put a name on a group of organisms so that we can communicate. So if we can use this as our definition, then everybody can be happy. Now, the problem comes in when maybe we use different criteria for emphasizing what makes a useful definition of a species. So some of these include things that some of you will be very familiar with, the biological species concept, which emphasizes the interbreeding properties of natural populations. This is a very good definition for vertebrates. It's not the greatest definition for many other groups of organisms. And so depending on the group of organisms that one studies, this might or might not be a good definition. There are other ones that emphasize things like the evolutionary history. And so you might want to make your grouping of organisms be something that is easily depicted on a phylogenetic tree. So this is another definition. This is actually one that I kind of like because it doesn't exclude any particular groups of organisms because of properties that they go through with their biology. But basically, one of the definitions, we share this with our students in our undergraduate taxonomy class, a species is what a good taxonomist says it is. And in all practice, it basically comes down to something like this. Of course, one has to put forward an argument and then have other members of the scientific community accept that. But really, this carries more weight than you might think. Okay, so we have a species. However we might want to define it. Let's just refer to it as a grouping of organisms. And how then does one group of organisms, is that derived from another group of organisms? And that is the process of speciation. So what is speciation? The origin of species. But what processes can generate new species? How can we actually get from one species to a pair of species to more species and so on? And how long do these different processes take? And what are the consequences? How do we get 400,000 species in 140 million years? And what sorts of processes and what sorts of consequences do those processes have? So I'm going to just review with you a few different ways in which new species can originate. And in all of these cases, these processes are not unique to plants, but these are some of the biggest and most important processes that generate new species of plants. So the first of these is geographic speciation, and we'll come back to ecological speciation and polyploidy in a few minutes. So if we think about ecological or about geographic speciation, there are a number of different ways we can do this. And one of the ways that I think is has very clear repercussions is if we think about very recent history in North America. So of course, North America has been covered by ice, and then the ice has retreated, more ice ages, more retreats, and so on. And so we can see here the maximum extent of glaciation about 20,000 years ago. But of course, if the ice is going down this far, cold temperatures are moving much farther south. And so what happened as a result of this glaciation is that there were huge movements of plants and animals and fungi and microbes farther south into different habitats. And one of the things that happened is that species became potentially isolated when the rest of the population started to move back northward. So as conditions got warmer and the ice sheets retreated, species began to move back northward. Now we have an excellent record of plants and their movement during these times thanks to the pollen record. So people who specialize in studying pollen have been very successful in telling us that, well, as far south as, the southern US and even down into Mexico, there are species that currently occur, in southern Canada and in the northern US. Now, what's happened more locally is that a number of species became actually isolated from the rest of their relatives as the rest of the relatives moved northward. So there are a number of species here on the Lake Wales Ridge that were actually restricted or isolated from the rest of their relatives. And over time, and again, this is not very long, maybe 20,000 years or so, there has been divergence of these populations from the relatives that moved farther north, to the point that we now have new species that are recognized in areas like the Lake Wales Ridge. We also have other areas of very high rare species occurrence in Florida. So in terms of plants, there are a lot of those in the Apalachicola region, and there are some other ones in the Big Bend and a few other areas sort of scattered around the state. But this region of the Lake Wales Ridge is really home to many rare species. And in fact, the local National Wildlife Refuge here was the first such wildlife refuge set aside for the protection of rare plants. Usually these sorts of areas are established to protect, for example, in this region, it could have been the scrub jay or some other iconic animals. But in fact, because there are about 40 different plant species that are uniquely found here in the Lake Wales Ridge, this National Wildlife Center was actually established to protect those plants. And so this just shows some of that diversity, some absolutely beautiful plants, these growing here just on very white sand. Now, in fact, all of the southeastern US, as in this area shown in green, was recognized about 10 years ago as a global biodiversity hotspot. And to be recognized as a global hotspot there need to be at least 1,500 species of so-called vascular plants, that is plants that have conducting tissue. So this would be flowering plants, gymnosperms like the conifers, and also ferns and some of their relatives. And the other part of that designation, though, has to do with there being a lot of degradation of the habitat in that area. So something like 70% of the habitat has to be degraded or have been converted from natural systems, along with the high presence of so many species. So we have here the North American coastal plain, a biodiversity hotspot. It was number 35, I believe, in terms of global recognition. So this is one of a very few such biodiverse regions on the planet and right in our backyard. And the other thing that we can do through studies of species distributions is identify regions like the Lake Wales Ridge here as being what we call an endemism hotspot. So this is an area, again, very high species diversity of species that are found nowhere else. Now, of course, if we traveled to one of these areas, like the Lake Wales Ridge, we would see not so many of these beautiful, iconic species that occur in the region, but we would see land conversion in many ways, and how all of these land use changes are leading to loss of species. So loss of habitat will mean loss of species in this area. And something like only 10 to 15% of all of the natural habitat in this area is actually still intact. Okay, so geographic variation can result from things like glaciation, pushing species into new areas, and then species migrating back and forth following these different waves of glaciation. But geographic speciation can also occur in other areas. And this is a fascinating case of closely related species that occur in Eastern Asia and Eastern North America. In fact, there are 65 genera. Probably everybody knows what a genus is. A genus is a grouping of species. 65 genera of plants that show this pattern, where they're closest relatives here and here, without anything now intervening across this upper region of Asia and upper northern region of North America. And we think that this sort of pattern originated because there was at one time a continuous forest across Asia and across North America. And as climates changed in the distant past, this was about 20 million years or so ago, these forests moved southward and they became isolated. So one example of species that exhibit this type of geographic variation is the dogwoods. So this is Cornus cusa, which is native to Eastern Asia, and Cornus florida, which is, of course, native to Florida and other parts of Eastern North America. So sometimes geographic speciation occurs on a very small scale geographically, like the Lake Wales Ridge and areas just to the north, and in other cases, much more broadly across a huge expanse of the globe. Okay, so geography can be a big driver of new species, but so can ecology. So there can be speciation that results from changes in water availability, soil type and chemistry, temperature, and so on. And the one example that I want to illustrate here is something called serpentine soils. Now, we don't have much in the way of serpentine in the southeast, but this is a huge driver of speciation in the northwestern part of the US and parts of Oregon and California. So serpentine soils are high in metals with magnesium, iron, silicates, all of these other chemistry. And what this means is that these soils are really bad for a plant to live in. So most plants can't hack soils like serpentine. As I mentioned, this is common in Western North America, but even though it's hard and very harsh for most plants, if a plant could manage to adapt to serpentine, that soil and that environment is wide open for establishment and maybe divergence and ultimately then speciation. So we see sometimes very different morphologies. Here you can see a little plant here growing on serpentine, and you can see that there are almost no other plants growing around that. It's just rock and sand. This is very common in the mountains in California, Oregon, and Washington. We can also have ecological speciation that takes place through pollinators. So pollinator choices, this might be getting at your question, Mary, pollinator choices can actually separate how plants interbreed with each other. So this shows here a species of the genus Mimulus, the monkey flower. This species is pollinated by bees, and this species is pollinated by hummingbirds. So these are close relatives, but they almost will never interbreed in nature because their pollinators are so different. So some changes in floral morphology, coloration, and so on will lead to attraction of different pollinators, and then those pollinators will drive can drive speciation with only plants that look like this mating with each other and those that look like that mating with each other. And there are a lot of other ways in which ecology can also drive speciation. Now, the last example that I want to share, and I'm going to spend a little bit more time talking about this one, is something called polyploidy or genome doubling. And here we can see a little diagram, and we'll be referring to this process as we go along. But basically what happens is, let's say we have species 1 crossing with species 2, and both of their genomes get retained within a single nucleus. And that will lead to a so-called polyploid species. And I'll give some more definitions in just a minute. But we can do the same thing here where two individuals of the same species actually will cross and produce what we call an autopolyploid. So basically, our typical diploid situation for ourselves, our chromosome number is 2n equal 46. So we are diploid. And the diploid condition is when the nucleus contains 2 copies of each chromosome. But it's very common in plants, and also we now know increasingly common in other organisms, that the nucleus may contain 3 or more copies of each chromosome. So this, for example, would be a tetraploid, this is an octoploid, and so on. Now, what happens is we increase in ploidy as we go from a diploid to a polyploid or even a higher polyploid, is that we have more copies of the genes in the genome. And that means there is additional material there for genomic innovation and also for then selection on new gene combinations. So just this process of polyploidy can lead to the generation of new genotypes, which will make new phenotypes. And those phenotypes then can be acted on by selection. So let's take a deeper dive into polyploidy. This is an incredibly important process in natural populations. It's a very common mechanism of speciation in plants. And as I mentioned, we're identifying more and more cases of where polyploidy occurs in other organisms. In addition, we now know that within a single plant, certain organs can be diploid and even have polyploid cells within them. And we see the same thing even in fruit flies and other organisms. So we no longer think specifically of polyploids referring to a whole organism. It could be cells or tissues within a single organism. And just as an example, I just mentioned that we're diploid organisms, but our liver, parts of our liver are octoploid. Parts of our heart are triploid. And several of our other organs are at least partially polyploid. In addition, our wound response. So if we cut our heel or something like that, the wound response often involves polyploid cells being produced because they're often produced, they're bigger, they make more product, and so they can lead to faster healing than if the cells were just diploid. In addition, many of our diseases we're now finding are actually controlled by polyploid cells. So many cancers are also polyploid. So something that we tend to think of as being a plant thing actually has a lot of application in other organisms. So both for agriculture, because most of our major crops are polyploid, but also in medicine. And so there's a lot of research now starting to trying to understand the polyploid nature of cancers. And we've started some discussion groups with members of the UF Cancer Center to see how we can bring together fields that 5 years ago we would have thought we would have had nothing in common to talk about in terms of research and teaching. But now we're understanding that, wow, what we've been studying in plants for decades actually has application to human health. The other minor point that I'll mention here, it's not minor overall, but it's minor at this point in my presentation, is that all flowering plants trace to an ancient polyploidy event. And I'll come back to that a little bit later. Okay, so I showed you a few diagrammatic examples of what polyploidy might look like. A haploid with one chromosome, a diploid with two chromosomes, etc. This is the species of the fern Ophioglossum reticulatum, and this is actually the adder's tongue fern. It has 1,260 chromosomes. It's out of control in terms of its chromosomal makeup. And we see this a lot in some of the ferns and their relatives, huge numbers of chromosomes. We don't have any good understanding at this point as to why their chromosomes are packaged in the way they are. Why do they have so many? It turns out they don't have any more genes than any other self-respecting plant, but they've got so many chromosomes. And so there's a lot of interest in just trying to understand what the basis and mechanisms of polyploidy might be and what are the consequences. Now for the last, let me see, I have to count this back, 25, almost 40 years, my husband Doug and I have been studying polyploidy in this genus of plants called Tragopogon. And this is a member of the sunflower family. And here we have three diploid species, Tragopogon pratensis, Tragopogon dubius, and Tragopogon porifolius. These three species are native to Europe, but they were introduced into North America in the early 1900s, and in particular into eastern Washington and adjacent Idaho. And in that region, these two diploids crossed with each other and then doubled their genome and formed this species that was named Tragopogon misellus, and this one named Tragopogon mirus. So these three diploids each have 12 chromosomes. These two tetraploids have 24. Now, this is an amazing example of very recent speciation. So as I mentioned, the diploids were introduced from Europe in the early 1900s. These three species, the allotetraploids, were discovered in 1949 by Marion Ownby, who was the director of the herbarium at what was then Washington State College. He named the species and described them scientifically and published their names and descriptions in 1950. So these are very recent species. We know from herbarium specimens, that is museum collections, that these diploids were not present in that region prior to the 1920s. So even if these tetraploids formed a little bit before that, it wasn't much before. So it's one of the few cases where we know without a doubt what the ancestry of these species might be. And we also know almost exactly when they were formed. And these tetraploids have formed repeatedly. Basically, every time two of these diploid species come into contact in a vacant lot somewhere in eastern Washington or adjacent Idaho, they cross and they form another population of the same species. And incidentally, these polyploids have never formed in Europe in the native range. So they've only formed, well, I will say only in the Pacific Northwest. Not quite true. There have been records reported from Flagstaff, Arizona, Sheridan, Wyoming, and somewhere, Gardner, Montana. But pretty much just in that region. So this just shows this in a little bit more detail. Here's this region of eastern Washington and adjacent Idaho, shown here, south of Spokane in particular, and this region where all of these populations occur. So this just occurs right along where that star is. Now, with these sorts of tetraploids, because that's what these are, two diploid species crossed with each other, we expect tetraploids to combine the features of their two parents. And so that expectation, if we're looking at chromosomes, would look like this. Here's diploid 1 and diploid 2. Here's their diploid hybrid, one of each. And here's the allotetraploid, where we're basically just combining parent 1 with parent 2 in a new genome. So this would be our expectation. And we had a postdoc working with us, Mike Chester, who was absolutely phenomenal in doing this sort of chromosomal work. And so he started asking the question, are the tetraploids really additive like this? Do they contain the contributions, equal contributions of the parents? Now, we would expect, because they are 12 and 12, that the tetraploids would have 24 chromosomes. But let's see what we got. Well, we actually found some individuals that only had 23. So they had monosomy here, just one chromosome of chromosome pair E that was contributed by this species. And then in this individual, there were 25, not 24. And here we have trisomy, where we've got 3 chromosomes also from that parental type. Now, those numerical, what we call numerical aneuploids, that means they don't have the expected number, they deviate from that, were not too surprising. What became really surprising was what have been termed in the literature compensated aneuploids. So here, they all have 24, but they don't have 12 and 12. So if we look at this individual, it's got 3 green copies of E and only 1 orange copy of E. So if you didn't use this particular amazing set of chromosome dyes, you would never know that you were deviating from that 12 and 12. And then if we look at this individual, this one's got 1 of B in green and 3 of B from orange. Again, the same thing. If you just used a regular dye, you would never see that. And then here, look at this one. This one's really amazing. All 4 copies are the green copies and no orange copies. So this phenomenon had never been described from natural populations, even though people had speculated that it should be out there. And we were blown away because something like 60% of the individuals of this trigopogon tetraploid had some form of this compensated aneuploidy, which means these individuals are not additive. They've got extra copies of this parental type, fewer of those, or extra of those, fewer of those, and so on. And this means that there's a lot of jumbling of the genome that's going on. So what does that mean biologically? Are these tetraploids additive of their parents? Well, we could see from chromosomes that the answer is no. But we also started looking in more detail at the genes that were present on those chromosomes. And we also see a lack of additivity. Sometimes there are 2 copies from one parent, no copies from the other. Gene expression is also not additive. So you'd think you'd get half of your gene expression from one parent, half from the other. But no, sometimes only one parental genome is being expressed and the other one is essentially just lying there. So what we see is that there's lots of novelty. And in fact, every individual that we examined across hundreds of individuals had different combinations of genes or chromosomes, so that no 2 individuals were exactly alike. And that meant that there is actually a lot of novelty being generated through this process. Now, let's take a deep breath. We're almost done. So what I've been describing here in Trigopogon is stuff that happens at the population level, just among a small group of individuals. And we would refer to this sort of process as being a microevolutionary process. But I think you can imagine that these microevolutionary processes that take place over generation after generation after generation eventually can be manifested in bigger, what we call macroevolutionary patterns. And so the types of things we see happening at the population level, like these genome duplications, the formation of polyploids, can actually be traced deep in angiosperm phylogeny. And we can do that because genomes show signatures of ancient duplications. Now, it was a huge surprise when the first plant genome that was sequenced and published back in 2000 to 2001, a tiny little plant with a tiny little genome, it was picked because it had a small genome. It was obvious right after that genome was published that it actually was multiply polyploid. It had a whole bunch of copies of genomes in it, even though it was this tiny little genome. And so that led people to start asking questions about genome structures in plants more broadly. And every plant genome that has been sequenced ever since 2000 has shown evidence of these ancient duplication events. So that's telling us that these processes that are taking place currently, every day, basically, in places like Eastern Washington, are showing up deeper in evolutionary time, and they're captured in the genomes. So one of these examples is back here. In this case, we've turned our phylogenetic tree on its side. So this is the present and the tips. This is farther back in time. And here we can see that in the what we call the common ancestor of all of the flowering plants, there is an ancestral duplication that traces back to that point. And that means that everything that came after that has a set of duplicated genes that all trace to that point in time. More interestingly, maybe, is that just about every one of these groups, every one of these branches is marked by another ancient duplication. And so we can trace the history of these polyploid events back in time through genome signatures. Okay, so what does that mean? You might think, okay, she's going a little crazy on this stuff because she must think this is interesting. But in fact, it's one of the ways that so many species have been generated. Polyploidy is a big, important mechanism of species formation in plants. What we also see is that all of these major groups, like the grasses, like the mints, like the bean family, all of them have been marked by at least one of these ancient duplications. And we think that reorganizing the genomes after these duplication events has led to the innovations that have spurred on speciation and adaptation in those groups because they've got extra gene copies. They've got stuff to mess around with. They can experiment essentially with one copy of the gene while letting the other one do its basic function. And this has all led to these really complex genomes. A lot of the diverse compounds that are used for medicinal purposes that have been identified in plants trace back to some of these ancient polyploid events where there are ancient duplications. And this also, we think, leads to a bunch of novel features. So there are a lot of angiosperm species. They've originated in a short period of time, and they have been generated through a diversity of processes like geographic speciation, ecological speciation, and maybe, most importantly, polyploidy. Now, the impacts of polyploidy, though, extend beyond the generation of plant species in nature. I've already referred a little bit to what we're learning about applications of polyploidy in medicine, for example, through drugs and drug discovery and also cancers. We also see application through biotechnology of generating new polyploids experimentally so that we can maybe harness some of the power of these extra genomes. All of our major crops are polyploid. A lot of the most complex processes on the planet we now are finding out have to do with genes that were duplicated by polyploidy. And of course, polyploidy is generating lots of biodiversity, plant diversity, animal diversity, even microbial diversity. So I'd like to thank a number of people who contributed to the work that I described today, most especially my husband and collaborator, Doug Soltis, also Matt Gitzendanner, a former graduate student of ours at Washington State University who moved to the University of Florida with us and who helped run our lab for over 20 years and who now is one of the chief leaders of the educational programs associated with UF's Hypergator computer cluster, and Mike Chester, whose beautiful chromosome work I mentioned a little bit ago. I'd also like to acknowledge the Polyploidy Integration and Innovation Institute. This is a collaboration of 9 institutions. We just got our funding from NSF last year. We're hoping it will stay. This is a 6-year program to compare the outputs of polyploidy in plants, in fruit flies, in yeast, and in any other organisms that our collaborators are able to sort of invite people in with. And thanks to NSF for funding. And finally, thank you all for your attention. It's been a pleasure to be here, and I'd be happy to take some questions. Thank you.
Turn this on. I'm going to ask a question and then open it to the floor. How do I phrase this? Is one of the reasons that you saw the polyploidy in the Northwest US due to the mechanism of pollination that was different than the mechanism in Europe?
Oh, perfect question. Yes. The diploid species occasionally overlap in distribution in Europe, but not very often. So there aren't a lot of opportunities for hybridization. But what we have also found is that in North America, the pollinators are generalists, and they'll travel between the yellow species, and they'll go back and forth between the yellow and purple species. In their native range, they have their own specific pollinators. And so there has to be a mistake for any sort of crossing to take place in Europe. But there's one place we're keeping our eye on it. There was a dissertation published in, I think, about 2003 from an island off of Stockholm where there are diploids and triploids, but no tetraploids yet. So they're diploid hybrids, and yeah, we want to kind of keep an eye on that. Yeah.
Questions? I was going to say there must be many questions. I've got like 10.
Clearly, there's an observation bias in how you can measure the polyploids and but do you think the rate of speciation is increasing? And if so, is this all a human effect?
Yeah, that's a great question. As I think it was about in 2006 or so, there was a really nice paper by some colleagues at the University of Texas who were commenting on the fact that there are fewer barriers to hybridization between species now because a lot of times our land use practices have destroyed some of the barriers between species. And so species might end up in disturbed habitats that neither species might have actually been in before, but because of our modification of the land, both species might end up in, and then they hybridize. They might just cross and hybridize like that, or they might actually form new polyploids. And the same thing's true with interactions with invasive species. So we have many more invasive species that just continues to ramp up. And a lot of times an invasive species will come in, get established in a, in a disturbed site that's adjacent to a natural habitat for a relative. And so we see a lot of examples of hybrids and new polyploids forming. So it does seem likely that the rate of speciation is increasing due to some of these processes and yeah, related to human activities. So yeah, great question.
Yeah, this is actually pretty similar, I think, because a lot of domestic plants are polyploid. So is that due to the domestication?
Yeah, so one of the things that is true of most polyploid cells and tissues and organisms is because they've got more DNA, the cells are bigger. And so then the tissues are bigger and the organisms are bigger. And if we're thinking about the domestication of any grain or other plant material, the yields are typically bigger, the fruits or seeds are bigger. And so they would have been unknowingly, that our ancestors would have been unknowingly selecting for polyploids because they were going for the greatest yield, the largest product or whatever. And so almost all of our main crops, that I had just listed a few on that slide, but almost all of them are polyploid, probably due to that size issue. So yeah, thank you.
It's very interesting, and I'm going to be building on yours next week. Um, Do you, on the question on speciation though, there are, and I'll be talking about that next week, there are places that are so isolated now from where they would normally maybe, the populations would be sharing genes that are now isolated and therefore speciating. And
Right.
It-
Yeah. So it kind of works in multiple ways, right? So things that maybe used to be able to breed more broadly and stay connected are now, like the Lake Wales Ridge endemics that got isolated and restricted in distributions and are now starting to speciate. So yeah, totally true. Yeah. So multiple mechanisms. Yeah.
I have two quick ones, hopefully quick ones. So polyploidy is apparently very important in speciation in angiosperms. How about other critters, animals and plants? And the second question is, why did something decide to become an angiosperm 140 years ago? I mean, obviously some mutation, blah, 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 but
Right. Yeah.
I mean, was there a niche that needed to be fit? Why did this happen?
Yeah. So I'll
It's
tell
pretty
you- yeah.
pretty complicated change of
It is a complicated change, and it's something that, so I just reviewed a manuscript yesterday for, one of the top journals in science, trying to get at this question. I mean, Darwin referred to the origin and basically diversification of angiosperms as an abominable mystery. And many people start out their papers today, Darwin referred to blah, 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 as an abominable mystery. And we still haven't answered it. We still have that same question. So, it takes a lot of changes on one hand to go from not making a flower to making a flower. And so when we look back through the fossil record, there are almost no transitional forms. And this is why it's so difficult to try to understand and solve this problem. But there are some things, if you start just thinking about like not what, we might think of as like a lily or a tulip or something like that today, but look at what the basic functions of the different organs are, we can start to see the accumulation of those functions in other groups of plants. So for example, a pine cone or a cycad cone has some of the same properties in terms of function as the parts of a flower. And so if we can kind of think in terms of reproductive function, we can see some sorts of transitions. But what happened with the origin of the flower is not just the structures, but the whole biology of reproduction, where you go from not just having a sperm and egg unite to make, the seed, but you've got this extra fertilization event that makes the endosperm, which is the nutritive tissue for the developing embryo. All of this stuff all happened like around the same time. And that, of course, none of that is preserved in the fossil record. So we have, and what we know from experimental work and also work from the fossil record is that the plants, those early angiosperms, they had greater protection of their reproductive organs. They had better dormancy built into their seeds. They had better dispersal mechanisms and so on. And so all of that really led to big advantages. So the hypothesis is, and of course there's no way to really test most of this, is that once the flowering plants got established, they were much better equipped to move into different habitats than what the gymnosperms, which were mostly trees at that point, what they were able to inhabit. So there, as you said, a lot of changes all happened about the same time, and that was pretty fascinating. Your first question about the diversity or the extent of polyploidy in other organisms. So we see lots of it in angiosperms, lots of it in ferns, a little bit in gymnosperms. So sequoia and sequoia dendron are hexaploids. No, almost no other tetraploid or polyploidy within the conifers. And bryophytes have a little bit of polyploidy. Algae have a little bit. Although we're starting to find more and more examples in algae or in bryophytes, people just hadn't been looking there. And then other groups, we see it in insects. A lot of insects now we're starting to find out are polyploid. Um, Frogs, fish, a few examples in mammals and birds, but not very much. And then even bacteria, though, are starting to be shown to have like many, many copies of their genomes. So, and fungi have them too. So, like,
So related to that, there's a question from the chat. Is there a limit to the number of chromosomes that a cell can actually pack, I guess?
right. Yeah. No, that's a great question as well. So yeah, I showed that photograph of the 1,260. There's a report of another species in that same genus that has 1,440, I think. Again, looks just the same. But those are pretty unusual cases when we think about how chromosomes are packaged and how DNA is packaged. So for many organisms, there are physiological limits on the size of the genome, in terms of what goes into actually making a chromosome, what the other components are. There's some really interesting work that a few labs have been doing on some of the environmental constraints on genome size. So to make DNA, you need a lot of nitrogen, you need a lot of phosphorus. And so if you're on nitrogen poor soil or phosphorus poor soil, you're going to have a hard time supporting a big genome. But if you happen to occur in areas that have a lot of those compounds, then maybe there wouldn't be as much selection against those. So we're trying to actually do some studies where we map out the distributions of genome sizes across the globe and look at that in comparison to soil chemistry to see if there is evidence for strong selection against big genomes in areas with poor soil. So there are also physical properties within the cell, like the cells can't get too big if they've got to go through cell division quickly. Sometimes the physiology of the organism comes into play. So there are a lot of potential constraints. And so the fact that we have any organisms with these big amounts of DNA is kind of surprising. And so then, they become kind of the exception. And then you wonder, okay, what's going on in those cases that allows them to become so highly polyploid? So that's a really good question. We don't know much about it. So, yeah.
I have a puzzle about the relationship of polyploidy to the rate of speciation. I can see from your talk, and it surprised me that polyploidy can lead to new species, and maybe pretty quickly. We had a talk last week by Ken Sulak about ancient fishes that, like sturgeon, that have not changed much over long time periods. And he said they were all polyploids. So
The conundrum here, right?
What else is going on? Are there ancient plants that are polyploid that have remained the same species over a long time?
Yeah, so really good question. And that comparison with the various groups of fishes, like the sturgeons and salmonids and catastomids, all have this ancient polyploidy. And there is an interesting sort of paradox, because what happens when a polyploid forms, because it has twice the chromosome numbers as its parents, it becomes reproductively isolated from its parents. And so if it can, in the case of a plant, self-fertilize, it can get established as a new lineage like instantaneously and become a new species. And so you have this whole process of very rapid speciation, as you mentioned. But once you're an established polyploid, and maybe, in the case of a lot of those fish groups, they're actually autopolyploid, which means they don't have 2 and 2 copies from different parents, but they have 4 copies that are all essentially identical. It becomes really difficult from a genetic standpoint. You guys heard from Charlie Bear earlier, right? So talking about mutation, right? It's really hard to fix a mutation if you've got 4 copies of something. So let's say even an advantageous mutation arises in this chromosome. Trying to get that to spread across all 4 chromosomes within an individual or a population is a very tricky thing. So it'd have to be, so essentially what happens is there's a buffering against change once that polyploid is established. So the speciation can happen very rapidly, but then downstream change can actually be a really slow process. So does that make sense? Yeah, okay.
All of that is fascinating. Thank you very much. And this is my first exposure to polyploidy. I took macroevolution, but that was mammals, basically. But you mentioned polyploidy cells in human organs. What does this imply about perhaps our relationship with Neanderthal, Homo neanderthalis, Homo denisovan? What does that imply? Anything?
So that's a really interesting question. I don't know about any of the tissues that have been extracted from any of our relatives, let's say, as to whether or not they've ever been able to look at gene copy number or do anything to infer whether there was polyploidy. So just thinking about that is interesting. But I think what it reflects is that polyploidy has a lot of beneficial aspects to it that have been repeatedly selected for across different groups of organisms. And so our parts of our heart are polyploid. They're parts that, are working extra hard, and that it increases the physiological rate and the output. By having the extra genomes, there can be, additional the additional copies can make additional products and so on. And the same thing's true with our liver. And then there's one other organ. What am I missing? Anyway, I'm missing another organ that is polyploid. But I think what it shows is that there is some functional similarity that may represent some common ancestry, but it may just be something that's been selected for repeatedly across different groups of life because it works under certain circumstances. So, yeah, thanks. So mitochondria are really interesting. So they're essentially the remnant. Well, the mitochondrial genome is the remnant of an endosymbiont, and so each circular mitochondrial genome is actually haploid. But what you have within a cell are lots of mitochondria. So mitochondria, chloroplasts do the same thing in plant cells, but by having lots of them, they get some of those same benefits of that I was just mentioning in terms of function by being polyploid.
One really quick last question. The southern magnolia, it's got beautiful flower, but I don't know if you call it the fruit or not. It sort of looks like a cone. So is it an angiosperm?
It is an angiosperm,
Ah,
but it
just an old one.
represents, yeah, it represents the flower structure of that's very similar to some of the earliest angiosperm structures, where there's all the parts are like inserted onto that cone. Yeah. Mm-hmm.
Thank our speaker one more time for a very stimulating talk.
Thanks. Thank you.
And you guys have a great week. And next week, we finish up with biogeography, island biogeography, specifically looking, I suppose, at speciation, but in